Assalamualaikum everyone, and today me and my group members are from group 18. Well, I'd like to present a question 11. These are my group members. First, we'll be given questions. I would like to brief the question. First, sex-linked recessive genes of Drosophila, which is V, CT, and CV, produce traits called vermilion, cut, and cross-venous. The cut females will cross the vermilion, cross-venous males and produce all white types females and all cut males in F1. When the females were test crossed, the results, including both males and females, were as follow: 766 vermilion cross-venous, 759 cut, 158 cross vinless, 140 vermilion cut, 85 cross vinless cut, 73 vermilion, 2 wild type, and 2 vermilion cross vinless cut. These are the questions. There are 4 the questions that they need us to solve.
Hello everyone, my name is NURUL binti WAHAB. My matric number is F25A1622. Today I want to present for part A. We need to determine how the genes were originally linked in the three hybrid parent. To do this, we first identify the parental class, which are the phenotype, with the highest frequency because they are produced without recombination. From the data given, 766 vermilion cross-wingless, 759 cut. These are the two largest class, so they represent the parental genotypes. This means that in the F1, three hybrid female, one at chromosome carry the mutant allele, vermilion and cross-vinous together, while the homologous at chromosome carry the mutant allele cut. Therefore, the original linkage agreement is V plus CV plus CT, where V for vermilion, CT for cut, CV cross-vinous plus for wild type allele. In other words, the vermilion and cross-vinous mutant allele were linked on one chromosome, while the cut mutant allele was linked with the wild type allele on the other chromosome. In conclusion, the original gene arrangement in the three hybrid parent was.
For question 11b, I determined the estimate the distance between genes by identifying the parental and double crossover class. The parental class had the highest frequency, while the double crossover class had the lowest frequency. By comparing this class, only the CT genes change position. So CT is the middle gene. Therefore, the gene order is V to CT, CT to CV. Next, I calculate the recombination frequency between V and CT. The total number of recombinant was 162 out of 1,985 offspring, giving a map distance of 8.60 centimorgan. For CT and CV, the total number of recombinant was 302 out of 1,985 offspring, resulting in a map distance of 15.21 centimorgan. Therefore, the final genetic map is a V to CT was 8.60 centimorgan and CT to CV was a 50.21 centimorgan. The total between V and CV is a 2. To calculate the interference, we first need to determine the number of observed double crossover and expected double crossover. First, identify the DCO classes. The two least frequent offspring classes are wild type 2 and vermilion cross-vinless cut 2. Therefore, the total observed DCO are 4. Next, calculate the number of offspring. See, the answer is 1985. From the recombination frequency calculation, the distance between V and CV is 0.152 centimorgan, while the distance between CV and CT is 0.082. The expected DCO frequency is 0.012546. The expected number of DCO is 24.7. Next, calculate of the C. You can see the formula, and the answer is 0.162. Okay, to finally calculate the inference, we use the formula. You can see the formula in there. We use this and get the answer 0.838. So the interference value is 0.838 or 83.8%. Thank you.
Hello everyone, my name is Ahmad Sufyan. My matric number is F25A1637. Next, I would like to solve this question B, calculating the expected result of double crossing over. To find this, we look back at the individual genes distance we calculate in question B. Then we got a probability of a crossover in the first interval, which is V to CT, is 15.21%. Then divided by 100, then we get 0.15 to 1%. And then we use the same method, which is distance of interval 2, CT to CV, which is 8.16%. Then we get divided by 100, which is we get 0.0816. Expected frequency of the double crossover individuals is calculated by multiplying the independent recombinant probabilities of both intervals. Using the formula which is DCO frequency of interval 1 times frequency of interval 2. Then we get 0.15 to 1 times 0.0816 equals 0.012411. Expected number of individual out of total sample. Then use the formula DCO number equals expected DCO frequency times total spring. And then we got 0.012411 times 1985 equals to 4.64 individuals.
Thank you.